Hello, good morning, everybody. We're back. We just took a quick 15-minute break, and I'm ready for our next speaker. And then we'll go to lunch after him. And so I am really thrilled this morning. I have not had the opportunity to meet our next guest, and I'm gonna do my best because I haven't practiced this morning. Okay, so Dr. Ramaswamy Govindan, I hope I did that okay. He's like, yeah, I did go right. Okay, good. Well, thank you for joining us this morning. It's a Saturday morning, and it's always a little bit of a fire drill to try and get things going on the weekend. But as you know better than probably as good as we do, that cancer just doesn't take weekends off. So Dr. Govindan is the Anheuser-Busch Chair in Medical Oncology and the Director of the Section of Medical Oncology at Washington University School of Medicine in St. Louis, Missouri. His main research focus is to understand genomic alterations such as KRAS in lung cancer, working with the McDonald Genome Institute at the Washington University School of Medicine and the Center Genome Atlas TCGA project, sponsored by the NCI, which does a lot of this. He studies molecular alteration in lung adenocarcinoma, squamous cell carcinoma, and small cell lung cancer. So thank you this morning so much for joining us. And we are looking forward to hearing from you and having you share your talk with us about the pipeline of hope as it comes to KRAS. Thank you.
Thank you, Terry. I'm sorry that I couldn't be there in person, but I'm honored to be part of this meeting. And I want to say before I get started, I enjoyed our earlier panel discussion from my very dear friend, Dr. Awad, and from from Daryl's great presentation and all these activities. You know, as a patient and a caregiver, the toll these illnesses take on the patients and the families, I think is undescribable. And I really want to commend you for not only participating in these clinical trials, and also organizing these groups and putting together these activities. So I really, really want to say my big thanks to all of you. Of course, without our patients, there won't be any clinical trials, there won't be any progress. So we owe them a lot. So today I thought I'll just talk to you a little bit about how we approach this, developing drugs, from a very different perspective than what Daryl talked about this morning. Essentially going back to really describing what is cancer, how cancer starts, and how we really try to piece out these alteration. A genome, to put it very simply, is describing all the genes in a cancer cell or studying them in a normal cell or cancer cell and studying them in total. And now we have tools and technologies to really figure out how we, parse out these alterations in all kinds of cells. So we'll talk about that a little bit, and then I'll put this in context with regard to KRAS, and then I'll talk briefly about the study I was privileged to lead along with a number of others, and that brought sotorasib to the market, to the clinical use. And then we'll talk a little bit about where this is going to go. And that's what I plan to do in the next half an hour or so. I have no conflicts relevant to this presentation. And I've been practicing a lot longer than Dr. Awad is. I've been treating lung cancer patients for the past 25 years, and my practice is exclusively limited to lung cancer. And, these are all my patients actually I've treated over the last several years. And if you see that, you can see these three CT scans, and basically what you see here is this is the tumor here that you see on all these three patients. And in fact, all three of them had lung cancer. All three of them had non-small cell lung cancer. All of them had what is called lung adenocarcinoma. Adeno means glands, carcinoma is cancer, so cancer starting in the glands of the lungs. 15 years ago, 20 years ago, we would have treated them all with the same chemotherapy. And this is exactly what we saw. This was a big paper presented at a big national conference describing 4 different chemotherapy regimens. And you don't have to be a scientist or a clinician to know what these curves are telling you, is that they are all, you cannot even see a difference between them. They all overlap with each other. And the results were quite dismal. And we did not appreciate how different these cancers were. We looked at them under a microscope, called them lung cancer, non-small cell type, and we went off with the treatment. But things have changed now. Now we know that even though they are lung cancers, non-small cell lung cancer, lung adenocarcinoma, they're all driven by different genes. And now we treat them very specifically based on the gene alterations to the extent we can. We are not there yet completely, but we are making significant progress in this area. And KRAS is one of them. So to go back a little bit, many of you asked these questions, and Daryl referred to this, and Mark talked about this a little bit too. So basically, every cell in our body has 20,000 genes, give or take a few hundreds. And by the way, we have more cells in our body than there are stars in the known universe, not in our galaxy, the known universe. And in fact, if you take a small area of skin about this size, we're talking about millions of cells. Cells are very small. And so each and every cell is controlled by 20,000 genes. And there are 20,000 genes in each and every cell, I should say. And perhaps a few thousand or several thousand genes are more active in a given cell. For example, your skin cells have the same 20,000 genes as your brain cells, but the skin cells do different things than the brain cells because different sets of genes are active in the brain cells as opposed to the skin cells. And there's a beautiful system that modulates the gene expression and that makes the cells do what they do. And in fact, as you may have heard, you can take a skin cells and change just 4 genes, and you can then drive them down to become a kidney cell or a lung cell. And this is how some of the research is done. And this is called the induced pluripotency of cells, and you can really modulate them. So these cells are what we call them very plastic. Based on certain gene alteration, the cells can turn from one cell to another over time. So basically, of those 20,000 genes, a few hundred genes have been identified as those genes which we call cancer-associated genes. And these are genes, if they get messed up, could potentially lead to cancer. I told you there are 20,000 genes in each and every cell, and not all 20,000 genes can cause cancer if they get messed up. And of those 20,000 genes, a few hundred of them, 300 to 400 of them, the number varies a little bit. And when they get messed up, they can cause cancer, and KRAS is one of them. And how do these cancer cells, get messed up, or genes get messed up? As Daryl referred to, there is a letter code. In fact, there is only 4 letters, A, T, C, G, much like the basic notations in a music scale. These refers to different bases, and they code for what are called amino acids, and that in turn codes for proteins. And the DNA is the mechanism by which the genetic information gets passed on. It's very durable. And the DNA gets transcribed into what is called RNA, and the RNA gets translated into what is called proteins. And the proteins are the business end, and they make this. For example, when you talk about KRAS gene, and the normal code is G, it stands for an amino acid called glycine. And then in this instance, the G gets transferred, miscoded to C, which codes for amino acid called cysteine. And the way the amino acids are, and they change the structure of the protein, and the protein conformation gets affected, and that's what leads to a lot of these problems. And so there are various ways the genes can get messed up. We won't go into the details. And one of them is where the letter code can be misspelled. Here you can see, as I told you, the G can get misspelled to C. And that is one type of gene alteration. And Daryl was talking about this amplification. Amplification, remember, we get 2 copies of the genes, one from mom, one from our dad. And so these normally have 2 copies of the genes, and in some situation, only one is more active, and I won't go into this detail, but essentially we have 2 copies of every gene in each and every cell, one maternal, one paternal. And so everything in our cell has to be just 2, and every gene will have 2 copies. So what happens in cancer is, through mechanisms we won't go into details, instead of 2 copies, you can have 8 copies, 10 copies. You've seen 25 copies or 50 copies of the same gene. If that happens to be a gene that makes the cells grow more and more and more, and the cells get cranked up quite a bit, and that's what over time leads to cancer. And that's the amplification he was talking about. Here is an example. You have two copies, one maternal, one paternal, and in a given cell. And here is an instance where you have not one, but there are 4 additional copies. So that's what we call amplification. And then remember, I told you of those 20,000 genes, only a few genes are active in a given cell. And how do these cells modulate the expression of some genes? And they actually, what I call epigenetic alteration, they silence some genes, they make them more active. And again, it's too much, we won't go into the details. These are called epigenetic alterations, and that's not very relevant today. In some instances, a piece of chromosome can get inverted and then can sit next to a different gene and then make the other gene more active. This is called rearrangement. And one of the great examples of this is the ALK gene you may have heard of, and that gets rearranged and that leads to activation of this ALK protein, and that leads to a different form of lung adenocarcinoma. As far as KRAS is concerned, we need to focus on these where the letter is misspelled, which is basically what is the major thing. If the G gets transferred and changed to D, that's where you get the G12D, and gets changed to A, you get G12A, and then the amplification we talked about. And what is fascinating is that when I started practicing lung cancer in 1999, we had only one chemotherapy regimen that had to be given in the hospital. And Dr. Awad was going to kindergarten at that time, if I'm not mistaken. And so over time, we have learned how to really study these genes, even go back 10 years. That time we actually presented this at a national meeting 10 years ago. We went back and looked at our database. It's an international database, and really looked at, asked the question, how many of these genes are altered that we know in lung cancer? We had studied at that point, just 10 years ago, only 4% of genes. Of those 20,000 genes, only 4% we had studied. But now, fast forward 10 years later, we have learned quite a bit. And a lot of this came from the breakthrough that happened with the human genome human genome project, which actually was done at Dr. Awad's place at Harvard University, Broad Institute, and Washington University, where I practice. Half of the human genome was mapped between the two institutions, St. Louis and Boston, and the other half was done in Houston and in London. And that led to really development of tools and technology. And this project took 10 years to do, 4+ major institutions, and cost the taxpayer up to $3 billion, BS and boy, to map one human genome, all 20,000 genes. Fast forward 10 years later, my colleagues, here you see Tim Lay and Rick Wilson, mapped the very first cancer genome of a patient with leukemia, a patient with cancer anytime in the world. First time we did this at a price of only for $1 million, and the costs have come down. Now we can do this for literally for $1,000, and that technology has expanded quite a bit, and we're able to now diagnose that very easily. And today, if your doctor sends you a tumor sample for gene sequencing, where they sequence not just one gene, but a few hundred genes, that all came from the advances made through these projects. And the Cancer Genome Atlas project that Terry was referring to during the introduction was a massive project that really mapped out over 11,000 tumor samples from various cancer types, sponsored by the National Cancer Institute, to map out all the genes in the tumor samples from patients. And that's a huge repository that has helped us investigators ask several questions. We have known about KRAS gene mutation well before all the cancer genome sequencing advances. But what we have learned over these 10 years is that the KRAS gene mutation is not the only one in a given cancer patient. It also occurs with other gene alterations, and we are beginning to find this interaction between these various gene alterations in the tumor cells. Even though the KRAS is a driver, others kind of facilitate the process, and we are trying to modulate them. I'll refer to that towards the end of the talk very briefly. So now we can not only map the genes, but we can map what we call the epigenome, the things that silences the genes, and we can transcribe, we can actually map the mRNA. We can now study the proteins. I'll give you a couple of examples. The cancer cells metabolize metabolize the nutrients very differently than the normal cells. Now we can study them as well. And that's, I think, going to be a major frontier in progress in the next several years in cancer therapy, in my view. And what we showed in the Cancer Genome Atlas project is that all lung adenocarcinomas are driven by some alteration in what we call the RAS pathway. RAS pathway is a major signaling pathway that really tells the cells to grow in response to various stimuli, or sometimes because the genes are messed up, they grow on their own, as in cancer. For example, you cut your skin, the skin has to heal, and that process is also driven by the same pathway and various members of this pathway. And so we really find that the lung adenocarcinoma is driven completely by mutations in various members. Not all of them are KRAS. KRAS is only one member of this pathway. But that's something we proved through this genome sequencing project. And I'm sure you've seen this, and Pasi will show you later on today some figures like this, where you can see this is a schematic representation. And as you can imagine, in a given cell, these are not color-coded and they don't go in very simple. This is oversimplified. These pathways are very dynamic and much more crowded. And these are the key members of this pathway. And this is the KRAS that we were talking about all day today. And there are various other members that so elegantly Daryl showed today, the switch and the volume control and everything. And but you don't have to remember the specific details, but it is a complex pathway with various members with a lot of feedback loop. And all of you are experts at this by now. The KRAS G12C is the most common KRAS alteration in lung cancer, fortunately, because we can target that now. And I'm very pleased to hear progress being made in other KRAS forms. And before we've always been eyeing this pathway to go after this, because it's been known for 25 plus 30 years now that the RAS pathways are very critical for cancer and very critical for lung cancer. And we have been approaching this indirectly through the various members downstream. And but unfortunately, we ran into toxicities. We saw modest improvement and not much really progress by itself. And in the KRAS mutant field, there are a number of trials are still ongoing in various combinations. We'll talk about that later on. But the real breakthrough came because of, not because of biology, because of medicinal chemistry. And this is a figure, it's very complex, even for us who do research to understand, unless you're a medicinal chemist. And remember I told you about the genes, and there you can talk about that codon 1, 2, 3, and we can talk about this 4 letters I mentioned, A, T, C, G. And when they get translated into proteins, the proteins are not laid out like your necklace or pearl necklace, and they're folded in a very complex configuration. And that is it's a very beautiful mechanism through which these proteins interact. And now we have tools and technologies to map out what happens if a particular gene gets messed up in a particular location. And it turned out that they found a pocket through which we can really find a drug that can go in, essentially turn off that from being in the on position to an off position. So it's a really tribute to medicinal chemistry that happened 5 years ago, and that really led to where we are today. And that's really is a spectacular breakthrough. And I'm sure many of you have seen that. And I want to show a couple of studies that we did with the sotorasib. This is an earlier paper that was published in the New England Journal of Medicine last year. And the point of that is that majority of the patients had some decrease in the tumor cells. And this we call waterfall plot. Basically, any decrease in the tumor size on this side, any increase on therapies on this side, this is not good, and this is good. And we don't count them as partial response or meaningful response unless it crosses a threshold. So that's why these are labeled here. But you can see that majority of them had some decrease in the tumor cells. And this, we call it colorfully spider plot, which really tells you how long these tumors are staying shrunk. And that's basically what it tells you. The flatter the line is, it's better. The longer, of course, is better. You can see a lot of these patients have control lasting quite several months. And then more recently, we published this paper that's really looked at the activity of sotorasib in lung cancer with this mutation. And this basically, we took a group of patients with advanced lung cancer who had had previous therapy, and they got the pill once a day. And 80% of them had some kind of a chemotherapy with or without immune therapy. It's a very typical population with an average age of about 63. And we had 126 patients. And about more than a third of them had tumor shrinkage. And in a small number, we saw complete disappearance of tumor. In fact, I'm very proud to say, and I'm very happy to say this, that the very first patient with a response to sotorasib came from one of my patients. And that time it was still not clear whether this drug would work. It was a very wonderful memory at the time when we had to contact the sponsors and let them know the tumor had shrunk dramatically. And so it was really a wonderful moment that I'll never forget. And then this study also confirmed what we saw in the previous study. The majority of them had tumor shrinkage, even though only a third of them qualified for a very strict definition of tumor shrinkage. But you can see bulk of them had some tumor shrinkage or not. And it's a different way of showing how long the tumor stayed controlled. You can see that it is controlled for quite some time. But one of the important things, a nice thing with this drug and this class of drugs, I should say, is that the grade 3, 4 are serious side effects that really, make them quite sick and need hospitalization, etc. They were a very small number. Even though 1 in 5 had some grade 3 reaction, most of them are not related to the drug therapy. And that really was long lasting. And you can see that the most common side effects is diarrhea and some nausea, and most of them did not require dose modifications. So, but one of the questions that we are trying to answer is, of those patients with KRAS G12C mutations, why do some respond better than others? What about those 37% of patients who have a good response, a great response? And that research is currently ongoing. And I told you earlier on that through all the genomic studies, we have really found that the cancer genes, lung cancer cells, don't have just one mutation. They have lots of other mutations. By the way, not all mutations play a role. Many of them are in the also ran category, and only some of them play a role. There's a ringleader, that's the KRAS here, and there are second tier leaders, and then a lot of them are just going in for the ride, and they don't really play a major role in the biology of these tumor cells. But we know that some of them do. And the 3 genes that repeatedly pop up with the KRAS mutant patient, gene mutated lung cancer, is a gene called P53, a gene called STK11, and a gene called KEAP1. And there is some evidence that if you have a STK11 mutation, patients don't respond very well with immune therapy. But we asked this question, does any of these key gene alterations present along with the KRAS G12C affect the response? The short answer is no. And it's a little too early for us to say for sure. And but the STK11 is something we pay a lot of attention to because of the historical association with its lack of response to immunotherapy in these patients, and it didn't really affect the response to these agents. And I still think it's a little too early for us to comment on this for sure, but we'll be looking at this and others over time. So the important message is that the sotorasib is just the beginning, and hopefully with the various different combinations and other drugs like adagrasib and other drugs, will have more options for our patients. And I'm sure you'll hear about this from Pasi later on in the day. There are other drugs that are currently being developed, and he'll talk about the drug he's been working on with Muradi. And so I left it up to him. And in my opinion, these 2 are wonderful drugs, great, very good drugs, and we need to make them great by finding various different combinations and identifying patients who respond better to this one drug or the other on various combinations. And I just want to say briefly in the next few minutes that where we are going with this. We also published a paper 2 years ago that showed that, I won't go through this, but I just would point out that when you give patients the sotorasib and the immune therapy, we bring in more immune cells in the tumors. The more dots means more immune cells, and you can see that here. And compared to vehicle treatment, which is a placebo, the KRAS inhibitor itself, sotorasib, brings in immune cells, and a combination really makes the immune system work, immune therapy work better. So the studies are currently ongoing with both these drugs, and hopefully the immune therapy and this KRAS inhibition would work. I also want to point out very quickly that we have moved on from studying only genes and the RNA. We are now studying proteins. Until a few years ago, we couldn't study the proteins completely in all of them in total. And we published 2 papers back to back last year and this year, where we studied over 10,000 proteins in the tumor cells comprehensively. And also the proteins get modulated, they get altered a little bit and that really make the proteins very active. We studied them as well. And I just want to highlight one thing, and I won't go through this complex graph in detail, but I'm happy to answer questions. But you may be able to see that protein called SOS1 that Daryl referred to was seen in significantly high number in a very active form in KRAS patients. And also we showed other proteins that are quite active here, raising the possibility that the other combinations we may have to go after in these patients. And these are from human samples. These are from lung cancer patients. And these are very complicated, state-of-art, up-to-date technology that was not there even 5 years ago. And all of them would tell us to look where to study in terms of biology more than what we already know. SOS1 work has already been ongoing. It's well proven. It just confirms that it's an important player here in human samples. And there are other things that we need to study in lab models before we really go after and study them in the clinic. And we also can now, using fancy technology, we can really figure out, remember I told you that the STK11 is an important player. We showed that if you have an STK11 mutation along with KRAS, the immune system is downregulated, and that's why the immune therapies don't work in these patients. We also found an explanation why that happens, and that really raises the possibility of combining them with various different combination agents. Perhaps one day we'll combine sotorasib with the immune checkpoint inhibitor with a new drug that can really make them all work better, and that's what we are going. I talked to you earlier on about studying all the genes in toto and all the 20,000 genes, and I talked a little bit about how to study these proteins that I gave you 1 or 2 examples. Now we can go one more step. We can actually take the tumor cells and just break them apart and study a few thousand cancer cells by using what is called a single cell sequencing technology. And then we can look at the proteins using special antibodies and what is called site-off, an example here, where each and every dot you see is a cancer cell. And we can really look at this and say, this cluster has immune cells of one kind. This cluster has immune cells of a different kind. This cluster has a bunch of cells that are slowing down the immune system from killing the cancer cells. And this cluster has all these cells that are probably coming in the way of how the immune therapy would work. And so we can really find out in a given tumor what the tumor landscape is. Now we can do what is called the spatial transcriptomics, where we can take the tumor and then look at them precisely and see what cells are sitting next to each other, what genes are driving them, what proteins are present in that. And that's going to take us to an entirely different level of combination studies. And that, I think, is going to move the field forward very rapidly compared to what we had done in the past. And that's all happening in human tumor cells. And there's a big initiative going on that both Harvard and Washington University and many other centers are very involved, called the Human Tumor Atlas Mapping. We're studying them from the beginning to metastatic sites and really finding out how to alter and how to go after these proteins. Dr. Awad presented beautifully a work on what are the mechanisms that lead to resistance after you get sotorasib, how the cancer cells find a way to grow despite having this active drug like sotorasib and adagrasib. And we'll be looking at the mechanisms of resistance, how the cancer cells stop responding to therapy. And that's another way of really making progress in this. And the Mirati's drug they talked about, we talked about the other G12D inhibitors. And I just want to say that as we move forward, we're going to find novel combinations to go with KRAS G12C patients. We're going to find the mechanisms of resistance and then go after them. We're also going to really move this forward, not just stay in metastatic disease, but move forward to locally advanced stage 3 patients, and then go on to early stage patients to cure more and more patients. And that's how we make progress. I'm very confident that this field is now going to move forward at a much more rapid pace than what had happened with the previous other targeted agents, because the tools and technologies have really moved forward quite a bit in the last 10 years. And I really am very hopeful about this. And I want to wish you all of you the very best. And I'm grateful to be part of this program. And thank you so much for your attention.
Whoops, I don't hear Armand. Okay, thank you so much for joining us this morning. That was fantastic. I do have a couple of quick questions for you. If you have a second, you can hang around with us. Okay, fantastic. Okay, first off, But some other biomarkers are approved for targeted therapies as first line of treatment. Why do KRAS patients undergo other treatments before receiving targeted therapy?
That's a great question. And, the way the clinical trial development or clinical trials work is that, when you have a new drug, like I told you, when we had this sort of asset, we didn't know whether it would work in human beings. So you don't want to take some patients who have some good options still in the frontline setting, chemotherapy and immunotherapy, and subject them to a drug that may not work. The sobering point, Terry, is that 91% of the drugs or the trials that we bring in cancer therapy, at least until recently, do not turn out to be promising or worthwhile to pursue, and our success rate is still very low. And so it is important that we don't take patients who have lots of other options, newly diagnosed, and deprive them of those things first. So we always start off in the second or third line. And then once it's promising, we move them to the front line. And then we move them to stage 3. And then we move them to stage 1. And that's how it works. But stage 1 and stage 3, it takes a lot longer to get an answer. And you want to show that a drug works and then move them. And that's a very gradual, understandable, scientifically sensible and appropriate option.
Okay, so we're just not quite there yet, is what I'm hearing, but it's coming probably.
Yeah, it is coming.
Yeah. Okay. Well, that's good. That's good. That's promising, right? That's why we call this pipeline of hope, right? Okay. And then I got another question from online, if I could, please. It says, Dr. Govindan, can you talk about the benefit of these targeted treatments not being cytotoxic, which could allow for patients to get to local treatment, surgery, ablation, and such with less risk and hopefully curative intent?
Yeah, it's a great question. So it's a beautiful question with multiple part answers, but I'll be brief in the interest of time. Cytostatic chemotherapy drugs have all one thing in common. They go after rapidly proliferating cells. So they don't really look for cancer cells. They go after any cells that are rapidly proliferating. It turns out that in our body, most rapidly proliferating cells are, the bone marrow cells and some hair cells and the cancer cells in cancer patients. So that's why the chemotherapy affects cancer cells more than your normal skin cells, for example. But what we have learned is that the chemotherapy is a blunt instrument. It is what we need to be is much more targeted to really go after the root of the cancer problem. And the great example is chronic myeloid leukemia. That's where this all began. And we were giving high-dose chemotherapy and bone marrow transplantation. It was very blunt, very toxic. When we found the key gene alteration, a specific drug going after that, people took pills and now they're living 20, 30 years, a normal life. And that disease is a bit different. It's got one gene as a major problem, whereas most cancers have a lot of other genes have come along with them. And so these kind of targeted therapies don't affect the normal cells. Typically, they go after the cancer cells. And regardless of whether you have chemotherapy or targeted therapies, there are instances we operate on patients with metastatic cancer. Great example is patient with a single lesion in the brain or a lesion in the adrenal gland. If they do very well, I do send them for operation. We all had patients who have actually been cured, even in stage 4 setting. And so it depends on the situation, the response to therapy, the location of the tumor, the patient. Eventually, I think we'll be doing more and more, if not surgery, a selective radiation of those tumors using this focused high beam called stereotactic radiation treatment.
Okay. And if you could just say that one more time, because I like people to hear that there are stage 4 patients that have been I hate to use the word cured because it feels so permanent and then it feels like a letdown, but certainly no evidence of disease, correct?
Excellent, Terry. I think you said it more beautifully than I could have said this. I think the cure it brings in the connotation, right? For example, if you have appendicitis, once you have an operation, you're cured. And in cancer, we always worried about saying this and letting people down. But do I have patients with stage 4 lung cancer 5 years later doing very well? Absolutely. With very little or no evidence of cancer, yes. Fortunately, those things have come because of immune therapy at this point. Targeted therapies, we have seen patients with EGFR mutation. I have a couple of patients who are now 7, 8 years later going on with stage 4 lung cancer out the same way. So with sotorasib and adagrasib, we have not had that length of time, but with some combination, I'm hoping that that will happen. Does it happen to everybody? No, it's a small number of patients. But the fact that it can happen is the big news. 15, 20 years ago, this would have been unthinkable. What was unthinkable when I began this research was giving somebody a pill. I remember sitting in a room when gefitinib, the very first oral pill we tried in lung cancer targeted therapy, we were designing those trials. Most of us did not believe that a pill would do anything for a cancer patient, like lung cancer patients. And but now nobody questions that. That's really says a lot about how far we've come. We still have a long way to go.
For sure. We definitely have a long way to go. And at the same time, I don't know how good it's going to look in the movies. It's not nearly as dramatic for them to like just show up and take a pill in the morning. But I'm all for making that a lot more of the sense of normalcy, that's how we get treated, right? And, yeah, I don't know. That's
Terry, I don't want to take up your time, but I do want to say this. I say this to my patients all the time. And I think we have to be realistic about things. Doctors should not overpromise and should not lead them, even in the spirit of encouraging people. But I think the fact remains is that increasingly we are seeing those patients living longer and doing better. And with science and tools and technology I described, we'll be able to understand why that happens in those patients. And that's what we call extreme responders. And the more we learn from those patients, then we can apply back and say why it did not work in others, and then go after that and move more and more into that column. And more realistically, I think we have become better in making cancer a chronic disease where you take a drug and lead a near normal life, much like hypertension and diabetes. And I think that's happening now. It's not even a future thing. And that's what we should aim for. And hopefully we'll end up curing many of them along the way.
Completely agree, because this is really what the whole concept is living with cancer, not just getting a cancer diagnosis and dying. We have to take what we've got, and we need to keep pushing the envelope, developing the drugs and getting them closer to us sooner. And I already know that they're already working on the next set and the next iteration of whatever it is that we need. Do we have anybody else have any other questions just here in the room? I'm going to check my phone, make sure I don't have anything else in here. Is there anything else to say? Oh, hang on. I've got something come up here. Okay, so what are the major challenges, still for you, doctor, what are the major challenges that researchers are facing in the development of these drugs at the later stages, considering that the complex nature and dependency of KRAS in normal gene growth?
Again, it's a great question, Terry. But I think what people have to understand, appreciate is that cancer, cancer is, aging and cancer are two sides of the same coin. The gene mutations are inherent. That's why we are where we are today, because the mutation is part of the evolutionary process. And you cannot eradicate cancer. You can decrease the cancer incidence just substantially by removing all the carcinogens and decreasing them. But I think the process is that it's hardwired into the cells. And the normal cell does not become a cancer cell overnight. It takes many, many, many years to get where it is. Not that we can diagnose them 10 years earlier, but then a normal cell, there are a lot of checks and balances in the cells to prevent a normal cell becoming a rogue cancer cell, which is a good thing. Otherwise, we'll all have cancer every very early on in our lives. And there are also very good systems in place from the immune system standpoint to kill these cancer cells at the early stage. And the way the cancer cells avoid and escape the mechanisms of the immune control are being understood better and better today. But I think it is a very complex process. And again, the cells will adapt. And that's how the organisms evolved in the macroevolution. And that's why the resistance is an inevitable feature of all these targeted therapies. And the way we have to do this is to really make these cells extinct, like the way in the macroevolution species have become extinct because they really have been deprived of the nutrients, have really been cornered into a sense where they cannot survive. And we believe that cancer cells can undergo such a process, but that'll come through very diligent, careful, complex research conducted over some years. And we have to do this without hurting. It's almost like you got to target, a bad guy in a crowded market, specifically. It's just not easy to do. And that's a challenge, the toxicities of these drugs. We know how to kill the cancer cells in the petri dish, but then doing it in human beings that are hurting the normal organs is a challenge. And that's where you got to proceed this very carefully. So time, patience, and a lot of diligent research. And again, participation of patients like you. Without that, this would not happen. And it's really the way to go.
Oh, yeah, definitely. And so I heard you bring up like the combination therapies as well. And we also had kind of a question along those lines. Could you please get your thoughts on mRNA using conjunction with immunotherapy?
Yeah, what people forget to realize is that we have COVID vaccine today. This is so effective, and that came from two things. Actually, the Human Genome Project I mentioned laid out the bare bones for analyzing and reading the genome sequencing. And the mRNA vaccines that are developed for this were initially thought and being developed for cancer. And this, because it's a very much more simple in the COVID situation, that got adapted very nicely. At the moment, it's a little too soon to comment how it is. The challenge with all this mRNA-based delivery is that, one thing is to induce an antibody against a COVID spike protein. That's a little straightforward. And in really going after the cancer cells, getting into the cancer cells, targeting them without affecting the normal organs is a bit of a challenge. But I think that's going to be overcome in some fashion or the other. But it's a little too soon to comment that it is going to work or not going to work. But it will be something that we will get there over time.
So it's going to take more clinical trials, is what I'm hearing.
Absolutely.
It's not where we need to be, but we're getting closer to where we should be. And, in the meantime, we're still. Okay, so do you have any other questions? I think we've like really worn this crew out. We're going to lunch right after this. And so you're just like, thank you so much. I have one simple, I have one request and I have a simple question for you. Okay, so here's the simple question. Do you have any theories as to why this has a tendency to hit GI issues when we're taking these drugs?
That's something. It's not unique to this unique to sotorasib or adagrasib, Terry. A lot of these oral drugs affect this, and the intestinal lining. If you think about the bowel wall, there is an internal lining, and this epithelium lining is shed literally every few days. And every few days, every 3 to 5 days, you have a new internal lining, and you don't even know that it goes out. And so these cells are rapidly turning over, and many of those cells need some parts of these proteins. And even though they are not mutated proteins, and this relining of this epithelial line intestine, I think it's messed up by a lot of these drugs. So that's my theory as to why the diarrhea is an issue with these almost all these drugs.
Okay, great. And then our one thing that we are doing with all of our amazing presenters today is we're asking you to do it with our group, the whole Cancers KRAS, which you can see back here on the screen. And so we're going to do it like y'all should be like pros at this right now. We're going to do like the whole 1, 2, 3 count, and then we're going to go Cancers KRAS. And we hope that you will join in with us too.
Of course, let's do it.
Event. Okay. All right. Here's a 1, 2, 3. Cancers KRAS. Thank you so much for joining us this morning.
Thank you.
Okay.